started, the response is also good. So what is this really potent substance? It turns out to be nothing other than acid, pH. Just changing the pH of the extracellular solution from a pH of 7.8 to 7.5 to 7.2. By 7.2, there is no more synaptic transmission. That's one of the reasons why it's so critical that your nervous system maintain a stable level of pH, because otherwise, at this pH, your pH is about 7.38 in your blood. If it drops to 7.2, you're close to death, as it turns out. So pH is really, really, really a potent modulator of neuronal transmission. Okay. Whoops. So now I'm going to show you where the glial cells come in. And that is going to be over here. So first I'm going to show you this picture here. This is a beautiful drawing by Ramon y Cajal, one of the fathers of neuroscience, in about 1890, 1900. This is a drawing that he did of the cell types in a retina. I don't remember what type of retina this is. It's probably a mammalian retina. Photoreceptors up there, second order neurons down here. They make synaptic contacts here. These are the ganglion cells, the output neurons of the eye. And this is one of those glial cells. And what I want you to appreciate here is this fine set of hairs, these fine fibers. These are the guys that are going to wrap and envelop virtually every synapse. And they also happen up here where, again, we get synaptic connections from the photoreceptors to the second order cells. So again, these glial cells are in a sweet area to be able to modulate neuronal activity. And what we have discovered, let me show you what one of these guys looks like when it's isolated first. So this is an isolated tiger salamander glial cell. Just got to do something here. And we're going to just go down. These are optical sections. And you see these very fine fibers here and there? These are the guys that are going to wrap and envelop. And this is after I've ripped the cells apart, right? So, I mean, there's still a lot of spaghetti strings on there, and I've probably ripped off most of them in this process when I isolate the cells. So again, these guys are going to be extensively intertwined with the neurons. Now, I want to go back to tell you why we are so hot to trot on the glial cells and on acid. And that's shown for you. Uh-oh, I did something bad.
I think you threw it in your garbage.
What did I do?
Wait, did you throw your Safari in your garbage?
I bet you that would be bad.
Click the
Trash?
No, like to see if it's on your bar still.
Wait a second. First, let's see what's in trash. Okay, you can do that. So I'll have to put Safari back. No, not yet. I don't think Safari's there.
It's probably behind all the screens.
Oh, so you think I just put it here?
Put it on your desktop.
I think I took it off. I gotta put it on again. The new Mac. Yeah. Okay. Still get used to the new Mac. Okay. All right, there you go. So here's the idea. Oh, I gotta tell you what we're doing. Hold on. So what we're gonna do here is we are going to measure the amount of acid coming out of an individual cell. This is an isolated horizontal cell. And what we're going to do is to put a pH-sensitive probe right next to it, within a micron. And what we're going to do is we're going to move that electrode slowly back and forth and sample the level of acid here and the level of acid there. And we're going to subtract this value from that. Yes, so we're going to find out, is this area more or less acidic than the point over here? This is a reference point. It's a this, okay? So that's what we're doing up there. We did it first with horizontal cells because there are some researchers who claim that it's the horizontal cells that spit out neurotransmitter, acid, and that inhibits nerve cells. But we know that that's not the case because this is the nature of the experiment. Here's one of those cells. We go there and back. This is the pH-sensitive licks, and what we're actually measuring is a voltage difference across this licks that's proportional to the level of acidity. Okay, so we're going to go back and forth, back and forth. And this happens to be a horizontal cell from a skate. This is a big cell. Okay, so now, when we're measuring from that cell, we see first it's sitting there and it's spitting out a certain amount of acid before I stimulate it. I'm going to do a control. This is a very important control. We're going to just drop in this about 1 milliliter of the same solution that the cell is sitting in. Shouldn't do anything. That ringer solution is the salt solution. I'm going to put it on, nothing happens. Now I'm going to put on the neurotransmitter that activates that nerve cell. That's glutamate. And when I put on glutamate, if those researchers are right, I should see a big increase in acidity, right? The horizontal cell should start spitting things out. And in fact, what we see is not a big increase, rather it becomes more alkaline right next to the cell, not acidic. So I think their hypothesis is baloney, right? This data says, no, when you activate the horizontal cell, it doesn't make things more acid, it makes it less acid. And we know the molecular mechanism by which that happens. And I won't go into it. So this is just a remainder of the trace. All right, so now I'm going to show you, though, what happens when you do the same thing with a glial cell. So here's the response from that horizontal cell that I just showed you. Now, we're going to do it with a glial cell. That's the red trace. And when I add something that activates the glial cell, you can see there's a big increase in acidity. You can't see it on the same scale, though. In order to fit it on the graph, I have to reduce the size of the signal from the horizontal cell down to this. And then, when I activate the glial cell, this is what you see. Boom. That's acid, right? So, this happens from an isolated glial cell from the tiger salamander. And, yeah. So, this is that same response, that big increase in acidity. This is a control when I move the electrode away. It should read 0, and it does. And then when I put it next to the cell, the signal is there again. What activates this glial cell, what activates virtually all glial cells, is extracellular ATP. On the inside of most cells, every cell has ATP in it, but in the inside of most cells, there's about 5 millimolar, 10 millimolar ATP. Look at how little extracellular ATP you need to be able to elicit an acidification. It's as little as 1 micromolar ATP. So these cells are really sensitive to small amounts of extracellular ATP. Yes? And if you increase the level of ATP, you get a whopping big acidification like you see here. So now we know that the glial cells spit out acid. And again, we also, what I showed you is acid really dramatically alters neurotransmitter release. Now I'm going to show you our hypothesis for how this is all happening. Okay. Oh, before I do that, I'm going to tell you one thing. So I've mentioned the fact that we see this from tiger salamander. is it unique to tiger salamander, or does it occur across a variety of vertebrate species? So what we did was to first begin with the lowest vertebrate that we could find. That's lamprey. And Simon Alford has lamprey around, so we were able to get eyes from lamprey. These are the kind of fish, animals that hook onto the fish in Lake Michigan and suck their guts out. Okay, so we took a retina, we enzymatically dissociated the cells. This is one of those retinal glial cells, and sure enough, when you add extracellular ATP, you get a big acidification. We've seen that in lamprey. We've seen it in a variety of different fish, like catfish, like zebrafish, like skate, shark, a few others. We've also seen it in glial cells that we have isolated from rat and mouse in the retina. We've seen it in a certain species of frog. We've also seen it, we got 2 eyes from 2 monkeys from the west end of campus, one macaque, and I forget what the other species was. We saw the same level of acid, and we've even seen the same from human glial cells, not donated by Dr. Murphy. I just put him here because he's fun. Okay. But we got these from the ibex. Someone had an accident, they died. 2 hours later, we had the eyes. We dissociate the cells, and they give a response. Kind of makes me a little queasy, you know? I mean, I know this person was declared dead. That said, the glial cells are alive. All right, so this then, on every vertebrate that we've looked, we get the same response. That means it's conserved across all vertebrate species, probably. That means that suggests that this is an important mechanism, that it should have been conserved through all of these different species, you know, over the course of evolution. Okay. So now I'm going to just tell you our hypothesis for what's going on. I'll show you a little bit of the data for it. Here's supposed to be a nerve cell. Nerve cells, you know, have vesicles filled with neurotransmitter, like glutamate. It turns out that those vesicles are also chock full of ATP. So when the vesicle fuses, it releases not only glutamate to activate the neuron, but the ATP comes out, which we believe activates the glial cell. The ATP binds to a special class of receptor. Do you guys know G protein coupled receptors at all? Maybe not. This is a special kind of G protein coupled receptor that responds to ATP. We know that because we can block it with drugs that block this receptor. So if we put in those blockers, there is no more acid coming out. When I wash away those blockers, the response comes back. So that tells us that this is probably a metabotropic G protein coupled receptor. We then think that activation of this receptor activates something called phospholipase C and inositol triphosphate. This is a guy that activates the release of calcium from intracellular stores. So first, let's block these guys and see what happens. This is a blocker of one of them, and you can see the response is much reduced. This is a blocker of the other one, and the response is much reduced. Okay. We think again that IP3 activates calcium release from the endoplasmic reticulum. We filled glial cells with a calcium reporter, and you can see that normally ATP induces a change in the signal indicating that calcium